So at this point, we know what homologous recombination is and how it functions. But the question is now, what can we do with homologous recombination? And this is a technology that we as molecular biologists can use to integrate new genes into genomes or plasmids or whatever we want. Basically, hijack the cellular machinery to allow for homologous recombination to insert or knock out genes as we are interested or as we want. So this was originally done in yeast, and the first example we're going to talk about is definitely the yeast example, and it has been done quite successfully in yeast and is quite a bit simpler to do in yeast than in higher eukaryotes. As you probably are aware, we can do this in mice, and we use this significantly for the purpose of targeted gene knockouts or gene disruptions or stopping genes from working. The first example is in yeast, and this is now using a kanamycin gene to replace a gene that we'd like to knock out. And so this begins by first doing PCR. And what we have here is primers for the kanamycin gene with overhangs that are homologous to sequences within the yeast genome. And that's the key here. So it's not just that we're PCRing our kanamycin, we're PCRing, replicating the kanamycin gene with a bit of homologous sequence. So we're engineering a new bit of DNA here, and what that winds up looking like is here where we've got our little homologous sequence and our kanamycin gene. And so when we put this kanamycin gene into our yeast, we wind up having some recombination here. And so this is to signify that there's recombination at these points. And what this winds up doing is putting in the kanamycin in place of your favorite gene's open reading frame. So YFG-ORF is your favorite gene, open reading frame. So we've replaced this gene with kanamycin. This is beneficial to us for a few reasons. First of all, we no longer have our gene that we wanted to knock out. So this is an actual knockout. We have completely obliterated the gene. The other thing is that we can select for yeast that have been treated in this way because kanamycin is an antibiotic. So the yeast that have had this recombination event will be resistant to treatment with kanamycin, as opposed to without the recombination event, they won't be resistant to kanamycin. So we're able to use that. So again, the key here is, though, these primers with some homologous sequence, and it's usually about 50 base pairs. So knockout or recombination by homologous recombination, knockout or disruption by homologous recombination in mice is a little bit more complex, but it can be done and is done frequently. And this was first done in the 1980s by Mario Capace and Oliver Smithies. They again made a gene using PCR. They PCR'd a gene with some flanking regions that were homologous to their gene of interest, the HGPRT gene. So again, this is homologous to this portion of the gene. This is homologous to this portion of the gene. And so they use this neo-R gene, and that's because this is a gene that, again, just like the kanamycin resistant gene, will confer resistance to an antibiotic. The antibiotic's name is G418, and that will allow us to select for the cells in which this homologous recombination has taken place. So when we have our homologous recombination, it's again via this region here and this region here, and we will wind up replacing the sequence in the middle. So we have 6, 7, 8, and 9. 6, 7 will replace 8, and use 9 for homologous recombination. So what we've done here is put in a neo-resistance gene, a resistance gene to the G418, and also at the same time we've interrupted or disrupted the HGPRT gene because it's no longer the full sequence, and so it won't be transcribed as a functional protein anymore. So we've effectively disrupted or stopped the production of this HGPRT gene by the insertion of this neo-R gene. So this allows us, as I said, to select for cells that have had this recombination event based on their resistance to this G418 due to the presence of the neo-R gene. But this doesn't help us in terms of knowing if there's random recombination occurring, because it's possible that this whole sequence could have inserted itself somewhere else. And so in order to deal with that, we have another mechanism called positive-negative selection. Positive-negative selection now is based it's pretty much the same as what Smithies and the other guy, I can't remember his name off the top of my head right now, figured out in the previous slide. So again, we have our neo gene here that confers resistance to G418. It's got homology to our gene of interest on both sides, and then it's got this HSV-TK sequence on the end, which is outside of the region of homology, and that's important. Okay, so when we have recombination, our recombination is going to be within these regions of, or in between these regions of homology. So for here, our homology is to this area, here our homology is to this area, and this is the area of the gene that the neo resistance cassette will replace. So when we have the HSV-TK, the reason that we use this is this makes a cell, when a cell is producing this protein, it makes the cell sensitive to another selector. This one's G418. Neo makes it resistant to the G418. The HSV-TK makes a cell sensitive to a compound called FIAU. So when the gene is inserted by homologous recombination, the cell will be resistant to G418 and sensitive to FIAU. So how that works is again by this. First, here's a diagram. Now the green areas are showing us the areas of homology. The red is not homologous to our gene of interest, and so recombination will take place in between these two areas of homology and will exclude this area that is not homologous. So when we have that occur, when the neo gene is inserted by homologous recombination, it takes place only with the areas in between these two areas of homology. So we get our neo gene here, and the HSV-TK is not included in this insertion. It is, however, possible that this whole thing, everything including the homologous regions, the neo-resistance cassette, and the HSV-TK section can be randomly integrated into the sequence of any gene without any homology. So for random integration, we could potentially have the insertion of this whole thing, including all of the homologous areas that in this case aren't even homologous to our gene, and the non-homologous area. So we get everything inserted. Okay, so by homologous recombination, we only have the insertion of the neo-R cassette, no HSV-TK. This means that normally, if there wasn't random integration, the cells would be resistant to G418 and not sensitive to FIAU. But in the case of this random integration, where we have neo and HSV, we have cells that are resistant to 418 but also sensitive to FIAU. So that allows us the potential to select by positive insertion of the neo-R gene and negative selection based on the HSV-TK. So any cells that have this random integration would die. Any cells that have the specific homologous recombination based on these sequence homology regions, only getting the neo-R gene inserted, would survive. So now we've talked about how to make our cells. different or change the genetic makeup of our cells based on homologous recombination. Now, how exactly do we use this to make a knockout mouse? And it's basically starting the same exact way. So we have embryonic stem cells, that's what ES stands for. They're going to be grown in tissue culture, so outside of the animal. We have our DNA with our altered version of the gene, and then we're going to insert our gene into our embryonic stem cells by homologous recombination. All of these cells are grown up, and only the cells that are, in our case, using the G418 and the HSV-TK model, we would select for cells that are resistant to G418 and not sensitive to FIAU. So we find the embryonic stem cells that only have the one copy of our target gene replaced by our mutant gene. We take these cells now, and we get a female mouse that is that has an embryo, and we inject our embryonic stem cells into that early embryo that's harvested from a female mouse. The stem cells go into the embryo, and then they are integrated. So the early embryo is partly formed from the embryonic stem cells. From that, we're going to take this embryo and put it into a pseudo-pregnant mouse, so a mouse that isn't actually carrying any embryos, but has all of the what's the word? All the hormonal changes associated with pregnancy, who is able to carry a embryo to term. We put it into that mouse. You wind up having some mice who have somatic cells that not all of them have our gene of interest changed, but some of them do. And so you then, over successive rounds of breeding, you wind up taking these mice and breeding a mouse that's going to be transgenic in all of its cells for that change of your mouse. So it's a big process, but it starts out with the same sort of idea of this homologous recombination being used to change the genetic makeup of your mouse, and then using, just breeding tactics to finally get a mouse that is 100% transgenic. So that's how you make a knockout mouse using homologous recombination as the starting point.